Hi everyone, Miss Mohern here to talk about what happens in eukaryotic cells to make proteins. DNA is transcribed into RNA, which is then translated into the language of proteins. So let's begin with the DNA, which is inside the nucleus of eukaryotic cells. Okay? Prokaryotic cells do not have a nucleus. And the first step is called transcription. So I'm going to go through that step, but first let's look at the DNA structure. It's a double helix, sugar, phosphate on the backbone, sugar, phosphate, and A pairs with T, C pairs with G. And you see one is right side and the other one is anti-parallel. Okay, so we got this going on. All right, so step one of transcription is that the enzyme RNA polymerase, which Jack will show the camera, is going to break all the hydrogen bonds. Go ahead, break it, run it, Jack. Go down, go down, go down. And it unzips the DNA, okay? So that's one of the jobs of RNA polymerase. RNA polymerase will then read and transcribe. So if it sees a T, it brings in an A. G goes to C, G, C, A, U, okay, T, A, and so forth. So RNA polymerase is transcribing until termination. And termination, we now have our immature RNA. All right, and this is happening in the nucleus. The DNA will go back. The hydrogen bonds will form between the bases. All right, so RNA polymerase now needs to be processed. So on the 5' end, it gets a cap. And go ahead on the end of that, look at that. What letter is that, Jack?
A.
How many A's do you see there?
I don't know.
A lot or a little?
A lot.
A lot of A's. This is called the poly A tail. Go ahead and add that to the end of the pink paper. Go ahead at the end. All right, so Jack is going to add that poly A tail to the end. So let's move it so we can see it. So we got a G3P cap and a poly A tail right there. Now, there's something else that needs to happen, and I will need some scissors, Jack. The introns are like intruders, and we got to cut them out. So let's go. I see some introns right here. I actually color-coded them a different color to make it easier on myself. And intruders, be gone. I'm going to use ligase to glue the coding regions of mRNA. Again, we need a cap on one end. G3P cap on one end of the RNA, and on the other end is a poly A tail. And I have removed the introns. Go by. All right. Now the RNA can leave the nucleus. So Jack, make it leave the white paper and go to the ribosome. So take this, chug it to this purple paper. Make it fit in between. Now let's look at what we have going on here. So this is a ribosome. There is a large subunit and a small subunit. Don't forget the poly A tail. Okay, so if we look at a ribosome, we've got 3 sites, EPA. Okay, now this is translation, and we can't translate from one language to another without a dictionary, so you will need your codon chart here. So here's what's going to happen. You're going to read every 3 bases together. Let me just get organized here. All right, so we're going to look here. These 3 bases are going to be read first. A tRNA will come in and bring in the amino acid that codes for those 3. The next 3 Okay, so every 3 letters are called a codon. And what the tRNA has is the anticodon, the opposite of what is on here. Okay, so if this is CAU, then the anticodon would read GUG. It's just the opposite of this. This is not what you're reading from. You're reading from the messenger RNA. And it's going to drop off amino acid, amino acid, amino acid. How do we join amino acids? Aha, dehydration synthesis, peptide bond, peptide bond. Okay, our mRNA is going to shift, our TNAs are going to exit, and we're going to keep growing this protein. Okay, so next tRNAs will come in. Okay, and the tRNAs have bring in a specific amino acid based on the codon. Okay, so dehydration synthesis. Goodbye tRNA. Dehydration synthesis, dehydration synthesis. Remember, that's when we lose a water, we lose an OH and an H from each amino acid and form a peptide bond. At the termination, when all of the mRNA is read, we are left with a protein that is in its primary structure. Okay, why is it in its primary structure? Well, because this is just the sequence of amino acids. What you can do to show, you can show the bonding for secondary. We have hydrogen bonds that form and coil the protein. Okay, and tertiary, we're going to see the R groups. So you got to look up, is valine hydrophobic or hydrophilic? If it's hydrophobic, it's going to hide on the inside. If it's hydrophilic, it's going to stay on the outside. So you're going to show this. I haven't shown this in my video, but the last part is for you to show primary, secondary, tertiary structure of this protein. And I hope that was helpful. Bye.